So in the last class, we had discussed the various enzymes which are used for manipulation of DNA. And in that context, we had discussed the nucleases. And within nucleases, we discussed about the exonucleases and the various types of exonucleases, DNases, RNases, and a DNA ligase enzyme, as well as certain enzymes which are used for DNA modification, such as the alkaline phosphatase or kinase, etc. So please go through that lecture again so that you are comparatively more aware that which all manipulation enzymes are used. So I had told last time that the endonucleases will be covered in the next class. So today we'll be discussing about restriction enzymes. And to my understanding, many of you might also already be knowing that what are restriction enzymes. So, but still there might be some students who are from different background, and maybe there are certain aspects of restriction enzymes that you're not aware of. So we'll discuss in the today's class. So it will go like that. First, we'll discuss what are restriction endonucleases, their nomenclature, the various types, which are the sequences that these enzymes generally recognize, what are the final products of the restriction enzymes once they act on a DNA. Then there is something called as isoschisomers and neoschisomers. We will discuss regarding the same, as well as the utility of these enzymes in certain scenarios. Then these are some general things, that how do you define a unit of restriction enzyme and which factors affect the restriction enzyme activity. And in my next class, I'll be discussing some of the artificial restriction enzymes that have been created or which are currently being used for genome editing purposes. I'll not go into too much detail because that will be, I guess, part of your genomics course, or there are certain other courses in which that is covered. But still, because we are, it's molecular biology, and you need to be updated with what's latest in a particular context, I'll just introduce you to some of these artificial restriction enzymes. Okay, so restriction enzymes, what are these enzymes? They are endonucleases. Okay, so it means that they make a cut in a DNA molecule, within the DNA molecule, and don't start to cut from the end, which is unlike exonucleases. One point is this. Second point is that how were these discovered, and why are they called as restriction enzymes? These are called as restriction enzymes because these were the enzymes which restricted the entry of a foreign DNA into a bacteria. So it was realized that in bacteria there are these enzymes, which were later called as restriction enzymes, which when bacteria is infected or some foreign DNA enters into bacteria, so to get rid of that DNA, these enzymes, they act on the foreign DNA. So then the question comes that how do the bacteria, they prevent their own DNA to be cut by these restriction enzymes? By marking them with certain modifications. What are those modifications? Those modifications are called as methylation. So what it does is it methylates its own bases in the bacterial genome, and mostly which bases they get methylated? It's the adenine residues. In bacteria, it is largely the adenine residues. They get methylated, and once they get methylated, so it means now it has marked its own genome and will not act on its own bacterial DNA. But any foreign DNA that will come, because it will not be methylated at specific sequences, these restriction enzymes will go and cut it. So this is how, through this restriction modification system, these restriction enzymes, they were discovered, and it was realized that there is presence of this restriction activity as well as methylase activity. So you can just go through how many enzymes have been identified till now, 3,000 enzymes, but I told you it's a dynamic field. Don't go by the numbers which are given. Maybe they have increased much more in number since maybe past 2 years also. And many of these enzymes, because of their utility in the field of genetic engineering and making recombinant DNA, because they allow us to cut DNA at a specific site, many of these restriction enzymes, they have been isolated from various sources. Generally, these REs, they are present in bacteria or in archaea. And from these sources, they have been isolated and further purified. And almost all the molecular biology labs will have these restriction enzymes. There are some restriction enzymes which are very commonly used in certain labs only because of the reason that the plasmids that are being used for cloning purposes, they do have a certain set of restriction enzymes which are present there. So comparatively, you will find that some of these enzymes are used in majority of the labs, while there are some enzymes which are used in only some labs in which some specialized work is carried out. So again, means if you want more details, you can go to NEB website, and very detailed overview of restriction enzymes is given there. Then what these are endonucleases. So which bonds do they cut? They cut the phosphodiester bonds between two nucleotides. And what is the final product of these restriction enzymes? They generate a DNA fragment which will have a 5' phosphate group and a 3', at the ends it will be 5' phosphate and a 3' OH at the point of the cleavage. So do these restriction enzymes, do they have any requirement for certain cofactors or temperature requirements? We'll revisit this, but the restriction enzymes that you use in the lab, which belong to type 2 category, they generally require magnesium. So in the buffer, which is used for carrying out the restriction digestion, it will already have magnesium in that. And because of the utility of these restriction enzymes, you can understand that all genetic engineering is almost based on the use of these restriction enzymes. It is Professor Arbor, Smith, and Nathans who got Nobel Prize for the discovery of restriction enzymes. Okay. So, how do you distinguish these restriction enzymes from the other nucleases that we had discussed in our previous class, which again were endonucleases? Some of those were endonucleases, like DNA is one, it's an endonuclease. But the restriction enzymes that we are talking about today, they differ from DNase 1 in the fact that these are much more specific in nature, because they go and recognize a sequence, a specific sequence in a DNA molecule, and then make a cut either within that recognition sequence or far away from the recognition sequence, depending on which type of restriction enzyme we are talking about. So the restriction enzymes, they have been categorized into 4 types, and these types are based on the fact where do they do they recognize and cleave at the same site, or they are recognizing and cleaving at different sites, or whether they have same subunits or a particular activity, and what are their cofactor requirements. So I'll just come to that particular table where you can see the distinguishing features about the various restriction enzymes. But yes, some facts are written here. So one is that they cut at a very specific sequence of bases, and they are the recognition sites, they are usually 4 to 8 base pair in length when we are talking about type 2 restriction enzymes. And cleavage occurs within or near the site. Again, this I'm saying in general for type 2 restriction enzymes. What is the nomenclature? How do you give the name to these restriction enzymes which are continuously being isolated from newer sources? So the nomenclature is like such. one very famous restriction enzyme is EcoR1. So E stands for the first letter of the genus, and species say COIR from coli, and these two they are written in italics at many places, and strain R, and order in which it is discovered. It means the first restriction enzyme that was isolated from E. coli R strain was EcoR1. So if you find another enzyme from this restriction enzyme from uh, E. coli with the R strain, then it will be named as EcoR2 and so on. Okay. So this is a very exhaustive table in which you can see that restriction enzymes, they belong to 4 different categories. The one that you use in the lab generally, which is mostly used in the lab, are the type 2 restriction enzymes, and then there are various variants of the same. So then let's talk about each one one by one. So type 1 are these restriction enzymes which recognize a sequence, but they cut very far from the recognition sequence, around 1000 base pair away from the recognition sequence. While type 2 are the ones which cut within the recognition sequence or very close to the recognition sequence. Then type 3 are the ones which have a recognition sequence, but they cut around 25 to 27 base pair outside the recognition sequence. And type 4 are also like type 2 in the sense that they also cut uh, close to or within the recognition sequence, but they generally act on methylated DNA and require magnesium to function. And these are just some of the distinguishing features which are listed here, but there are much more. There are many other features that can distinguish these 4 different types of restriction enzymes. Another important point is that I told you that in the restriction modification system, system, which is present in bacteria, there is that methylase activity which will methylate the adenine residues which are present in the bacterial genome, and then there is endonuclease activity which will act on the foreign DNA. So in type 1 REs, you will find that endonuclease and methylation activities, they are in the same protein. But in case of type 2, it only has endonuclease function. And another point is that type 1, for example, they require ATP, and the donor of methyl group, which is SAM, which is S adenosyl L-methionine. So it requires both ATP and SAM to function, while it requires only magnesium to function. So you can just go through all these points. It's very difficult to memorize it, but for your simplicity, I can say that the restriction enzyme that you are currently using in the lab for cloning purposes, they are type 2 restriction endonucleases. I have given a big arrow to type 2S as well, because in my next class, when I'll be talking about artificial restriction endonucleases, in that context, I'll introduce you to one enzyme, which is type 2S type. Okay. And then, yeah, so these two are the major ones which we'll be talking about. Now, one question actually came in my mind, that we keep hearing that restriction enzymes, they cut only double-stranded DNA. So frankly, just before the class, I had these questions in my mind. Do restriction enzymes, they cut single-stranded DNA as well? So I came with these a few exceptions. So there is one study in which they have shown that some of the restriction enzymes, they can cleave single-stranded DNA as well. So such enzymes, some examples are given here. But again, I want to point out that nothing is hard and fast rule in molecular biology. You may find newer and newer enzymes being identified, which may be exceptions to the rest of the category of the enzymes that we are talking about. So this is some exception. Again, I'll say that all the restriction enzymes that you are actually working in the lab for cloning purpose, they are specific for double-stranded DNA only. But among this whole battery of REs, there are some which can cut single-stranded DNA. Then I had this question, are there some REs which have been identified which can cleave the RNA? So again, I found that in 2010, they had done some screening studies and in a DNA RNA duplex, and they found that some restriction enzymes which are listed here, which belong to type 2P class. I told you that type 2 has multiple classes. So type 2P class, which can recognize a palindromic. What is a palindrome? And I'll just come to that. But then they can cleave uh, the DNA or the RNA molecule which is present as in the form of a RNA DNA duplex. Okay, so they did this study and then they found that what is the final product, and they found that in some cases some enzymes were cleaving the DNA in the RNA DNA hybrid, some enzymes were actually cleaving the RNA in the RNA DNA hybrid, and some enzymes were making a clear double stranded cut. So means I thought that it will be good that you get aware with the same. And very recently, last year, there was a follow-up study of these restriction enzymes which and cut RNA as well. And they found that the restriction enzymes that cleave RNA DNA heteroduplexes, they bind double-stranded DNA in A-like conformation. Means they did some follow-up study and they wanted to understand that uh, which conformation makes these enzymes cut at in certain context, and then they said that probably it's the A-like conformation. So I've given the names of these papers so those interested can go through it. And you can understand that while we do have these machinery or these scissors to cut DNA at a specific site, we don't yet have the ability to cut RNA at the desired site. So in that context, these enzymes are very important, and we are actually looking for enzymes which can cut RNA at a specific sequence just like DNA, and we are able to ligate two different RNA molecules together. So that's why it's good to keep working on identifying enzymes which are maybe specific for RNA sequences and making a cut there. So now coming to the recognition sequence, I told you that generally it is 4 to 8 base pair, and mostly these recognition sites, they are palindromic in nature. What is a palindrome? It means that if this is the upper strand, this is the lower strand, they both will read same in each direction. From here to here it is GAA TTC, and from here it is GAA TTC. Okay, so this sequence is called as a palindrome. So in this case, it is when both strands are considered, it reads same from back and forth. Now, another thing that I want to tell, that for some restriction enzymes, there is a clear-cut recognition sequence that is given. While for certain restriction enzymes, you will see that they do have certain N in between. It means that they can cut a sequence in which GAATC is there, or let's say GAT, means any of the nucleotide can be replaced here, and still this particular enzyme will make a cut there. So it means that there is little bit ambiguity, or let's say little bit wider range for these enzymes to cut a particular DNA molecule. So this is about the recognition sequence. Then coming to what are the product of these sequences. So in that context, let me come to the blunt or the sticky end concept. So some restriction enzymes, they cut symmetrically and leave blunt ends. For example, see this particular thing. It is a DPN1 enzyme. There is SMA1 enzyme and many other enzymes. which make a blunt end cut. So in that context, what they do is that they make a cut on means symmetrically at the same side in both the strands, and as a result the final products that we get, they do not have any overhangs and they are blunt ended in nature. Okay. Then there are some endonucleases which cut, their cuts are not directly opposite to each other, and because of which they generate staggered ends. So in this context, for example, BAMH1, a very, very frequently used enzyme in the lab, it makes a cut immediately after G. So what happens, what will be the final product of this particular enzyme when it cuts on this DNA molecule? You will have G here and CCTAG here. So it means that this particular enzyme is generating a 5' overhang. Similarly, in case of PVU1, which makes a cut between C and T, it will generate a 3' overhang. So please, means whenever you are practicing at home, please make these structures and see that if you are able to see which enzyme is making a 5' overhang or a 3' overhang, and so on. So now if I, in a molecule which is cut with, let's say, PVU1 enzyme, I want to make these, the product of these enzyme blunt ended. So which enzyme will I use here? Let's say I have in a tube, I had DNA molecule and I cut it with PBU1. So it means that I do have in a tube the fragments which have a 3' overhang. So let's say I want to make this particular fragment blunt ended, this fragment. So how will I make it? What enzyme will I put?
And those exonucleases which cut the 3'
Again.
overhangs, and the exonucleases which specifically target the 3' overhangs.
Okay. So what I understood, you are saying the exonucleases that can cleave the 3' overhangs, right?
Yes, sir. Yes, ma'am.
Yeah, exactly. So, or you can use a T4 DNA polymerase, which can which does have a very good 3' to 5' exonuclease activity. Even that will make it blunt. You are right. You can use any exonuclease or a 3' to 5' T4 DNA polymerase also, which has a very strong 3' to 5' exonuclease activity. So, and in case of BAMH1, if you want to make this molecule blunt, what enzyme will you prefer? Any guesses? Am I audible?
Yes,
So you have these recessed ends. So how will you make it blunt?
clean off fragment.
Yes, you can use a cleano, means any DNA polymerase which can fill in the ends. Because in this case, you cannot add a polymerase to fill in the ends because polymerase always goes in 5' to 3' direction. It needs a free 3' OH group to actually add the new nucleotides. But in this case, it doesn't have a free 3' OH group. Here it is 5' phosphate group. So here the polymerase will not work, but here the polymerase will work and it will add the nucleotides next to each nucleotide complementary, and then it will make it a blunt end molecule. Now, coming to certain categories of these restriction endonucleases. So one is isocodamers. So they are nothing but the enzymes which produce same ends, same overhangs. For example, the restriction site for BAM-H1 is GGA-TCC, like I had showed you before, right? So in this case, when it is cut by BAM-H1, it cuts next to G, and what overhangs am I left with is GATC, right? So these are which overhangs? 5' overhangs, right? I'll just bring it here. These are 5' overhangs. Anyway, so BAM-H1, the overhangs are GATC. In case of BGL2, its recognition site is different. AGA-TCT. It cuts after A. So what overhangs I'm again left with is again same, GATC. Then I have another enzyme, SAW3A, in which the recognition site is GATC and it cuts just before G. So in this case also, the overhangs, the sticky ends that are generated, they are GATC. So in case of all of these enzymes, even though their recognition site was different, the ends generated, the sticky ends generated by dilation from these enzymes, they were same. So as a result, if I have a molecule which has been cut by BAM-H1 and I have another molecule which is cut by BGL2, can I ligate these together?
Yes.
We can ligate it together because the sticky ends are the same. So in case, let's say I want to ligate this fragment with the BGL2 generated fragment, what will happen? It will NNG, then GATCTNN, and similarly CATG will combine with this here. So, right? Means I hope you are getting it that these two fragments, they can easily combine together because they have the compatible overhangs and which can bind to each other. So now my question is, similarly BAM-H1 and SAW3A1 cut fragments can also come together because the overhangs are the same. So this fragment can easily ligate with this fragment, right? Now, my question is that if I have a molecule, BAM-H1 cut molecule, which I combine with BGL2 cut molecule and ligate it. So as a result, I have a new molecule being made, right? Now, if I want to cut that molecule with BAM-H1, do you think I'll be able to cut that molecule?
The palindromic nature has been destroyed, so it won't be cut by either of the restriction enzymes.
Exactly. Very rightly put. Because now see, if I combine BAM-H1 cut fragment with the BGL2 cut fragment, what will be the final sequence that I get? It will be GGA-TCT. So it means it is not a recognition site for BAM-H1 anymore because now it is GGA-TCT, and similarly it is not a recognition site for BGL2 anymore. So it will be, I mean, in case of BGL2 also, the site is completely gone. So as a result, the ligated molecule will not be cut by the original restriction enzymes, uh, in this case BAM-H1 and BGL2. So all these things you will come across when you have to set up ligations and you have to decide that which enzyme you have to cut and how will it be ligated together. So in many scenarios, in practical terms, you come across situations in which case, for example, one, let's say vector only has a BGL2 site, while in case of insert, you have BAM-H1 site. So if it was, let's say, BAM-H1 site in vector and insert has eco-R1 site, you could not have ligated them together because they are not compatible with each other. But BAM-H1 and BGL2 are still compatible with each other and you can ligate them. So actually when you work in the lab and you have to make decisions regarding cutting and ligation, in that scenario, these type of information is very, very useful. Then
Ma'am,
we
I have
have,
a question.
yeah.
Ma'am, the question was, as you mentioned, that in the laboratory we can do such a thing that in which the BGL to and a BAMH1 cut sites, they can be ligated together. So, ma'am,
Right.
when these two cut ends they are put together in the same mixture, how do we ensure that that these two different ones they are ligating with one another and not they're ligating back to the, since they have the same thing, that they're not religating back?
So in that case, you will learn it in the practical for cloning to ensure that the molecule does not ligate back to itself and to prevent self-ligation or ligation with its own molecules. So you do alkaline phosphatase treatment. Why you do that treatment, etc., will be discussed in detail in the practical.
Okay, ma'am.
अगर तुम ये सोचो, अगर वेक्टर मॉलिक्यूल, लेट्स से इट इज कट विद BAMH1, if it doesn't have a 5' phosphate group, then it will not ligate to itself.
Okay.
Okay.
Yes, ma'am.
Probably, yeah. So when that practical is happening, we can discuss this further.
Yes, ma'am.
Okay, so another set of enzymes that we'll discuss is isochisomer and neoschisomer, and these are the categories. Let's say isochisomer are pair of restriction enzyme that recognize the same recognition sequence. For example, SPH1 and BBU1. Why their names are different? Because they have been isolated from different bacteria, and but they were found to have exactly the same sites of recognition, and they cut within that recognition enzyme site. So it means that they are isochizomers of each other, right? Then within a category of isochizomers, there is another category called neochizomers, which recognize the same site, for example, SMA1 and XMA1. So they both are recognizing the same site, but one enzyme is cutting, let's say, after the third C, while another enzyme is cutting after the first C. So as a result, their products and their overhands will be different. So again, I'm saying that isochizomers, they are the enzymes, restriction enzymes, which recognize the same recognition sequence and make a cut within the same. And within that category, there can be, let's say, two categories, one which cut exactly at the same site, and another category called neochizomers, which cut at different sites, and various examples are given for the same. Okay. For example, in this case, there are 4 different enzymes which have same recognition sequence, and see, they both, all 4 are cutting at different sites. So I'll revisit these isochizomers when I'm telling you about one of the utility of the isochizomers. Then the frequency of restriction, there is a formula by which you determine that a particular restriction enzyme, how frequently it will cut in a DNA molecule, considering that the GC content is 50% and 4 different nucleotides are present in equal proportion, and that too in random fashion. So this is a question generally put in various these entrance exams and all. So the simple formula is that a tetranucleotide sequence, for example, should occur every, so the thing is 4 to the power n. So every 4 to the power 4 is equal to 256 nucleotides, and a hexanucleotide, ideally, if these parameters stay same, that is, the nucleotides are present in a random fashion, the 4 nucleotides are present in almost equal proportions, only then this final calculation holds true. But indeed, it gives us an idea that a 6 base pair cutter means the recognition sequence of which is 6 bases. It will cut every 4 to the power 6, that is 40 and 96 nucleotides. So this is the formula how it is done. And however, in practice, it's not always true, because we all know that in a particular genome, any genome, these parameters might not always hold true, which is that you don't have a higher proportion of GC, or the nucleotides are, it's not always that nucleotides are present very randomly. So there are certain regions in the genome which are very GC rich. So in that case, even if I'm, calculating for a hexanucleotide which binds to a 80 rich region, then my calculation will be wrong. So that's why, considering these points stay same, this is the formula for doing it. And then there is something called as restriction mapping, which you can do as self study from the TA Brown book. It is very easy, that how do you map the, and I'm sure majority of you have done it in molecular biology course by Professor Preeti. This is a simple definition. 1 unit of RE is defined as the amount of enzyme required to digest 1 microgram of the appropriate substrate in 60 minutes in 50 microliter reaction under the conditions specified for that enzyme. So please bear in mind that all enzymes may not have exactly same conditions. The enzyme you're working in the lab, you should check in its brochure from the company, that under which condition, what temperature that enzyme works, has an optimal activity. So when you have to set up a restriction enzyme reaction, what things do you put in the tube? Obviously, you'll have your DNA, which needs to be cut. Then you put your restriction enzyme. Then you have a buffer. And in that buffer, I told you that while majority of the enzymes are magnesium requiring, in certain other cases, there can be certain other cofactors which will be put in the buffer. And then when you run it on gel, the digested DNA, so how long do you digest, again depends on which enzyme you're working on, but generally within 1 hour or 2 hours you will see digestion taking place. Some are quick working, some are not, so depends on which company you're buying and what conditions they are saying. But in general, in the lab we put for 3 hours. And so this is undigested DNA that you can see. And or let me first show you that how does a plasmid DNA look on gel. So if I have an uncut plasmid, I will see maybe a one band or maybe one two small bands for nicked or linear DNA. And if I cut it with restriction enzymes, for example in this case with EcoR1, then I see one band this and one for this. So here I'm able to see two bands here. And if I'm talking about a genomic DNA, I mean that too from, let's say, even from bacteria or human genomic DNA or plant genomic DNA, which are extremely big in size. So if you cut that with restriction enzyme, you will never see clear bands. What you will see is a smear of bands, a continuous smear of bands, which you will see here. So do you think this is, let's say, degraded DNA? No, it's not a degraded DNA. If a genomic DNA gets degraded, what you generally see is bands here at the bottom of the gel, which are like random degradation of DNA. So then you are sure that, oh my God, my DNA has got degraded. But if you digest it with a restriction enzyme, generally you see this continuous smear starting from above. So what do these each bands here represent is the size of the fragment that is generated by the restriction enzyme when it acts on the genomic DNA. And because genomic DNA is very huge in these cases, you will have all a continuous gradation of the sizes that are generated. And this is how generally your genomic DNA will look like after digestion. Then coming to which are the various factors which affect restriction enzyme. So while most of the restriction enzymes, they work very well at 37 degree, there are few restriction enzymes which actually work at lower or higher temperature. So that's why what I'm saying is that when you have to work with any restriction enzyme that is given to you in the lab, please see in the brochure that what temperature does it work. So SMA1, for example, works at 25 degree. So let's say if you have to put a double digestion, one enzyme at 37, one at 25 optimum, then it's better to do sequential digestion. You digest with one enzyme at 37 degree, then again you add fresh enzyme, the new enzyme, and then you set it up at 25 degree. So these are some practical things that you will understand when you actually work in the lab. Again, for the cofactors, I've already told you that apart from magnesium, some enzymes may have certain other requirements which are given here. And here also magnesium and sodium and potassium. All these notes will be shared with you anyways. Then buffer. So majority of the enzymes were found to be active in the range of pH 7 to 8. And also there is a possibility that restriction enzymes are working well. You have a DNA which does have a restriction enzyme recognition site, but what you will see that restriction enzyme is not able to cut it. Why? Because it was found that, let's say, even though the sequence was GGATCC, or let's say for BAMH1, but it was unable to cut at that site because, for example, the C residue was methylated. So depending on the methylation status of the adenine or cytidine residues present in a particular DNA sequence, there is a possibility that certain restriction enzymes will not cut. At the same time, there are certain restriction enzymes which will only cut if the DNA is methylated. Okay, so this is a very important factor that I'll just tell you in more detail, that depending on the methylation status of the DNA, a restriction enzyme may or may not cut at that particular sequence. There are certain restriction enzymes which do not care whether the site is methylated or not, and they will anyway cut. But there are certain restriction enzymes which will not cut if it is methylated, and there are some which will only cut if it is methylated. So please keep this in mind. Okay, so see, many times when we are doing epigenetic study, you are likely, you might be aware already that in DNA there are certain sites called CPG sites which get methylated. Or let's say, if I want to tell in more general terms, in DNA there are certain sites, sites with representation of certain nucleotides which do have a tendency to get methylated. If those nucleotides, they are methylated, and those sites, they are present in the promoter of any gene, then it will lead to gene silencing. It is generally seen that DNA methylation of the promoter, it is correlated with the silencing of the downstream gene. Okay, one point is this. Now let's say in many scenario we want to know that what is the methylation status of that promoter, whether it is methylated or not. Or let's say a particular DNA sequence, we want to know whether it is methylated or not. So in that scenario, it is good to use, or means we can use these isoschisomers. So these are those enzymes, both of which means they recognize the same recognition sequence, but they may or may not cut if it is methylated. For example, HPA2 and MSP1, they both are isoschisomers, as they both recognize the sequence CCGG when it is unmethylated. But when the second C here, when it is methylated, only MSP1 can recognize it, while HPA2 cannot. So if you want to differentiate between whether a particular DNA fragment has methylated residue or not, then what you can do is that you can isolate it, and in one case you will get a band. If you, for example, if you cut it with MSP1, it will always cut. whether it is unmethylated or methylated. But HPA2 will not cut if it is methylated. So if you compare the restriction enzyme pattern of in that particular DNA fragment, and you want to know of the methylation pattern, you can assess it by using isoisomers. Okay, now, okay, so this also becomes important when you are doing cloning. So, because there is a possibility that because of certain methylated residues, your enzyme is not able to cut there. So you please read this example here, and this is a easy example, and it is just saying that this EXPA1, its site is TCT-AGA. Okay, this is the site at which EXPA1 it makes a cut. This is the recognition sequence of EXPA1. Now, if this particular site it is followed by TC, then it which might happen when you have any vector, or when you have vector insert combined together. So what new base that we get is GATC. It is underlined, see here it is there. So this GATC is the site for methylation by the methylases which are present in the bacteria. So what will happen that in this scenario, the methylase, it will methylate this fragment, and as a result, now if I want to cut it with EXPA1, it will not be able to cut here. So you please read this whole thing, and there is also a link to the same. So just to understand the importance of the interplay between methylation as well as restriction enzymes. So another very important point, and probably the second last point that I want to discuss with respect to restriction enzymes, is that when you are setting up a reaction with restriction enzyme, you also have to be aware of the fact that it's not always that if the restriction recognition sequence is present, the restriction enzyme will always be able to cut it very nicely, because it is also dependent on the fact that restriction enzyme should get enough space to actually sit on that recognition sequence and make a cut. So if you can see that if the DNA fragment has only one base pair on either side of this particular recognition sequence, then you can see that even if I give a 20-hour time for BAMH1 to cut this DNA fragment, it will cut only with 25% efficiency. Even if my buffer condition and temperature condition, everything is optimal, because it did not get enough space to actually sit and make a cut. So in NEB or maybe in other companies booklet, you will get this form of chart, in which the companies, they have standardized, they have seen that okay, if two bases are added, then how much percentage cleavage is there, and if three bases are added, then how much percentage is there. So this becomes very important when you are actually designing primers for cloning. So when you are designing primers for cloning, you generally put a restriction enzyme site, because you want that you cut your this PCR amplified product with the restriction enzymes which are compatible with the restriction enzyme sites which are present in the cloning vector. So as a result, in those cases, it's very important that you put minimum 3 bases before your restriction enzyme sites. And for example, in NDE1 enzyme, you can see even if there are 6 bases next to it, then there is zero activity. So it needs 7 bases minimum to make a cut. Okay? So this is just to sensitize you that when you design primers and you put a restriction enzyme site, it is very important that you put certain number of bases preceding it because here on this side anyway your amplicon will be there, so you don't have to worry about this side, but this side you have to put extra bases. So when you put these extra bases, again depending on which enzyme you are putting, you have to see whether you put whether two bases are sufficient or three or four and so on. Last point, you means from practical point of view that you will come across is something called as star activity, in which restriction enzyme, it starts means cutting at non-specific sites as well. And generally what conditions have been shown to promote star activity of restriction enzymes, which is not desired, of course, are, for example, if the pH is not proper, it is more than 8, then it was seen that certain restriction enzymes started having star activity. Or in some cases, if in the reaction mixture you have high glycerol concentration. So as you might be knowing that these restriction enzymes, they are stored in glycerol. Okay? So many students, what they do is that, okay, my restriction digestion will be very good if I add more restriction enzyme. So that is never desirable. आपको जितना restriction enzyme चाहिए, you should put that much only. If you put extra, so what it leads to is high glycerol concentration, which either will interfere with the activity of your restriction enzyme. Maybe it's not optimal, and also it will promote the star activity. So all these, increased incubation time, some people say वो अपना restriction digestion, they set it up and then leave it at 37 degree for maybe 2 days. But it's never desired, because there is a possibility that some non-specific cleavage takes place. So these points are very important to keep in mind, because when you are setting up a reaction, this restriction digestion, and let's say you are again and again seeing that, ma'am, my ligation is not taking place, or my DNA is not digesting, I'm seeing extra bands and all. So these you need to troubleshoot what wrong did you do. So it could be you added extra enzyme, it could be that you had done it for increased incubation times and so on. So please keep all these points in mind. That's all with respect to restriction enzymes that I wanted to convey. I've already told that restriction mapping you can do it as self-study from T.A. Brown book, and rest all is given in the notes here, or the links for the same are given. Okay, so any questions? Okay, so
So when you talk about the single stranded cleavage of from restriction enzymes, does this happen